Good morning, everyone. It's 10 o'clock EST Eastern Time from South Carolina. My name is Dil Tavaraja. I'm a professor at the Clemson University, oversee the Organic Pulse Breeding Program and Nutritional Quality Program. And our program is funded by the OREI NIFA project and South Carolina Department of Agriculture, and also the Global Food Shot and the USDA ARS. Today is our April webinar. Actually, this is the last seminar with the speakers, because next seminar in the May, you will hear from this young scientist. But today you are going to hear from one of the prominent plant pathologists and the agronomist based in Williston, North Dakota, Dr. Audrey Kalil. Since her early career in 2024, she served as an agronomist and outreach coordinator at the Horizon Resources Cooperative, where she focused on mostly improving crop health and managing educational programming for growers in North Dakota and Montana. Prior to she joined the Horizon Resources, she was a professor at the North Dakota State University, and she was there from 2015 to 2024. And her research, again, that's where I met Audrey. Audrey was a young professor there when I was working, and she was primarily focused on diseases and regional staples like drumbeat, peas, lentils, and chickpeas. Audrey has a PhD plant pathology from the University of Wisconsin-Madison in 2015, and her BS degree is from University of Minnesota. She also highly active figure in the pulse grower community. She served voluntarily as the associate director for the Northern Pulse Grower Association and co-producing the Growing Pulse Crop podcast series. So she is doing a really great podcast that related to pulse crops. So with that, thank you, Audrey, for joining today to share all the research. Take away. Thank you.
Great. Thank you for that wonderful introduction. One point of clarification, at NDSU, I was a plant pathologist, not a professor. Our Research Extension Center roles are non-tenure track, but that was just fine with me at the time. So I'll just dive right in. The goal of my talk today was I'll highlight a few specific diseases, but in general, I wanted to highlight some strategies. So hopefully whatever you're trying to manage in pulse crops, if you're a first-time grower or a researcher just starting to work with pulses, whatever you're going to be dealing with in your location, you'll have some ideas of how to manage that. Let's see. So thank you to the powers of AI. I generated this diagram cutaway. There's a lot of things that we're managing in pulse crops, and today I'll be specifically talking about peas, lentils, and chickpeas. And again, that might differ by your growing region, but generally speaking, we have our above ground diseases. Here I've illustrated powdery mildew, which can be problematic, primarily on peas in our area. White mold, which if you grow dry beans or soybeans, you're familiar with that pathogen. It can be problematic in lentils in our area. And then asclepiated blight, that's a disease that can affect peas, lentils, and chickpeas. The pathogens are specific to each host, but is most problematic for us in chickpeas. And there might be different management strategies for all of our foliar diseases, but some things might benefit all of them, like crop rotation. And then below ground, these are pathogens that live in the soil and attack the roots or developing seedlings. So Dr. Lyndon Porter last seminar talked at length about managing Pythium. Today I'll be primarily focusing on Aphanomyces root rot, but there are other pathogens that can attack below ground. Fusarium and Rhizoctonia, those are fungal pathogens in general. But again, there's a lot of things we could talk about, and what you're dealing with might not be what I'm dealing with here in North Dakota, Montana, Northern Great Plains. But hopefully you'll walk away with some strategies to manage whatever it is you have going on in your operation. So, we're gearing this towards a system where chemical control might not be an option. And if you're looking at the concept of integrated pest management, that's where, we're using every strategy at hand to manage pests. This strategy was developed out of problems with insecticide resistance. So where chemical control was no longer working, we had to find other strategies. Obviously, if you're in an organic system, chemical control isn't an option. And that's where your agronomic practices really form much of your disease management program. And I really like this figure here. I didn't make it, but I found it. It's the base of your integrated disease management program. And much of it is focused on just preventing disease from happening in the first place, or from pathogens becoming established in a field or on an operation. Because once that problem occurs, then walking it back and managing it from that point on is much more difficult. So these are kind of the general things I'll be talking about: seed health, field selection, variety selection, and then planting date in particular, but there might be other agronomic practices that might manage whatever disease it is you're dealing with. So if you're not familiar with this resource, I encourage you to use that QR code to go to this website. The Regional Pulse Crops Diagnostic Lab is a very unique resource in the United States. Anyone can send a sample to this lab. And they have various tests for chickpeas, lentils, green peas, and yellow peas. They can test for viruses, nematodes, fungi, bacteria. So if you're a new pulse grower, or you've saved a seed lot from last year, I highly recommend you make it a standard practice to send your seed in for pathogen testing, as well as germination, seed weight. If you send it to this lab, you can check both options. But even if you're buying certified seed, they might test for some things, but not everything. So I would recommend sending seed in every year for pathogen testing. So one example of what we're managing with pathogen seed testing is ascochyta blight on chickpea. So this is the disease cycle of ascochyta blight. And I've highlighted here that seed infection can essentially start that disease cycle. So if you've never produced chickpeas before, say it's new to your region, it's essential that you have seed that's free of this pathogen, because that's how it's entering your field. Once that seed germinates, you have an infected seedling. That infected seedling forms we have these lesions. That's where, if you can see here, you have this reproductive structure, pycnidium, within that lesion. That produces spores that goes on to infect new tissue, and this cycle will just continue throughout the growing season. And so if we eliminate this factor here, right, then you're a much better chance of reducing disease risk over that season, and then you have less pathogen in that field in future years. So this is what that looks like. You can see this leaf lesion here, those black dots, those are the pycnidia. And so that will, under wet conditions, rain will splash that to fresh tissue. The spores will transfer to fresh tissue. You have more lesions. You can end up with lesions girdling the stem, and so then that whole part of the plant can lodge and die. And then it can end up looking like this plot right here. So that's a non-treated plot that was inoculated under irrigation. So hopefully no one's field ends up looking like that, but it can. And so make sure you're testing your seed and avoiding infested seed lots. So this is some information from the Saskatchewan Ministry of Agriculture. Many of these thresholds aren't strictly enforced. If you're growing certified seed, there might be limits for, say, ascochyta blight on the seed. That's state by state, and that's up to that regulatory agency in the state on what threshold they'll allow. So again, that goes back to making sure you test your seed and understand how much pathogen is on the seed. You can see, here for chickpeas, for Ascochyta rabii, which is the pathogen that causes Ascochyta blight, the economic threshold is 0.3% on seed. So essentially above that point, you're creating unacceptable levels of disease risk. It says do not use seed. That threshold might be different in another state. It might be 0%. So whatever state you're in, they might have different rules about the amount of pathogen that can be on a certified seed lot, but those rules aren't going to apply, obviously, if you're not buying certified seed or you're just saving seed, right? You don't know what's on that seed. If you're in an organic system, I would probably shoot for 0% just because you don't have a lot of tools, right? I have seen fields that have planted seed over this threshold and disease onset is very early. And really the only thing you can do at that point is have a very strong fungicide, foliar fungicide program. So there are other thresholds. So economic thresholds, sorry, I should have explained that. It's essentially the point at which you're creating a loss due to that pest, whatever it is. For field pea, it's 10% on seed. You need to use a seed treatment. So again, if you can't use a fungicide seed treatment, maybe you want to consider a lower economic threshold. If you just Google Saskatchewan Ministry of Agriculture guideline for seed-borne diseases of pulse crops, you can pull this up. There are various publications available that help explain how these thresholds work. They're not strict thresholds. You'll have your own, choice of what you want to do on your operation, but just something to consider not introducing a pathogen to your field to begin with on the seed. All right. So let's talk about field selection. Not every field is going to be very suitable for pulse crops. The soils we have here in North Dakota range from, very heavy ground in the east side of the state to sandy, rocky soil. Generally speaking, a pulse crop is going to like a well-draining soil. It's not going to like to sit wet. And it's not necessarily just because they don't like wet feed and there's poor oxygen and it's supposed to die from that. But a lot of our diseases, in fact, all of our fungal diseases essentially need moisture to go through their life cycle, to infect, to spread, etc. So where you have created compaction due to driving on the field, or it's just a heavy soil, maybe that spot doesn't drain well, it's sodic, and so the water just tends to pond on top. These are areas that are going to create disease in your field, and then it will spread throughout the rest of the field. So when we get to the concept of crop rotation, one field with a heavy soil might need a much longer crop rotation than a field with a lighter soil because of disease. So we can't treat all fields the same, and not every field is going to necessarily be suitable for pulse production. Here on the bottom here, I just show the reproductive structure of the white mold pathogen. You can see that soil there is really saturated. You need saturated soil conditions to cause that sclerotia to germinate. And then, that's what launches the spores onto the plants. So, in an irrigated system, we want to make sure those soils are draining and we're not constantly keeping the canopy wet, keeping the soil wet. So water management can play a role as well. If a field is otherwise fit, maybe consider tile drainage to improve drainage on that field for pulse production. But just the thought that not every field might be suitable. I'm not going to talk about herbicide history at length, but we've seen a lot of issues from carryover of certain herbicides, especially where you're not getting enough rainfall to allow that compound to degrade in the soil. Again, if you're in an organic system, that's not necessarily going to apply, but make sure you're keeping records of your herbicide applications. So, we're using more residual herbicides now to control our various resistant weeds that we're struggling with, and that complicates weed management. but it's just really important you keep records. Even 2 years back, you need to know what's been sprayed in that field to make sure you're not causing crop injury, which then makes the plant more susceptible to disease. I'll talk about crop rotation a little bit more at length, and then there's a soil-borne pathogen testing to determine if a field is carrying certain soil-borne diseases which can affect pulse crops. So I'll just give a little bit of a case study on Aphanomyces. I'll talk about root rot management a lot. As I discussed at the beginning, there are multiple pathogens that can cause root rot, but here in North Dakota, eastern Montana, what we're really struggling with is Aphanomyces. The full name, Aphanomyces eutyches. It can also attack alfalfa. So if you're an alfalfa producer, this pathogen can reproduce on that crop. There you have the option of Aphanomyces resistant varieties. We don't really have that option in peas and lentils. This pathogen won't affect chickpea, so that's an option in fields with big problems with Aphanomyces, but it can be devastating on peas and lentils. And once this pathogen becomes a problem in a field, it's an oomycete, and that means it produces these oospores, which you can see in this root cross-section here and down here. And these are thick-walled structures that can persist in the soil for many years, 10 years or more. And so if you grow peas and lentils very frequently in a field, you build up the amount of this pathogen. If you think about it like a weed seed bank, and at some point in time, the pathogen levels become so high, you can't economically produce peas and lentils in that field anymore. We have some tools available to manage root rot, but prevention, you really want to prevent it. So if you're a new pulse grower, having a good crop rotation to start can help you grow pulses much further into the future. So when I talked about, field selection and managing compaction, things like that, this is just an example of a pea field I visited a couple of years ago. On the left here, this is the compacted field entrance. And you can see the peas are very thin. They're starting to yellow from the bottom up. If you dig them up, they look like those roots on the previous slide. So you can see this compaction has kind of allowed the water to sit there, and then that makes the pathogen reproduce and spread. And it needs that moisture. And then on the right here, this is just a low spot in the field. So again, where water accumulated, these plants germinate, start to grow, but then the pathogen infects, and it can proliferate and spread in the saturated soil conditions. And the issue with that is maybe not your whole field doesn't look like that, but if you have a really wet spring, a lot of rainfall, then it can start to spread from these foci in the field. So you can see this is an aerial image of a lentil field in northwest North Dakota, and you can see it's starting to travel wherever water spread in that field, the pathogen starts to spread. And over time, with wet springs, it can spread through the whole field. And I've seen that occur where a huge section of the field might be completely wiped out. So we don't want to get to that point. So this was a study I initiated when I worked at the NDSU Research Center. We had a field that was planted to peas in 2016, and it tested positive for Aphanomyces. The whole site was planted to safflower in 2017, and then we started these crop rotation treatments. So on every other year of lentil and durum, that's very tight, but that has been practiced in some parts of our region for a long time. So that was kind of the base level rotation, and then 3-year rotation, 4-year rotation, and 5-year rotation with non-host crops. So small grains don't host this Aphanomyces, and neither does canola. I tried this lentil mustard intercrop. One farmer in our area intercrops his pulses with mustard, and he feels that helps mitigate losses to disease. Whether that's economically because you have mustard thriving in areas where lentil doesn't, or because he feels like it helps reduce the root rot. That was kind of what I was wondering and looking at. We had tried like a half season mustard cover crop. We're looking at the brassica species specifically because they've been used as a soil fumigant. They grow them as a cover crop and incorporate them into the soil, and the plants produce a compound that's toxic to fungi. We don't do a lot of tillage in our area. It's very dry. We don't have a lot of topsoil, so no-till is pretty standard in our area. So I was looking to see if an intercrop would kind of meet that purpose. Again, I tried a half-season cover crop, but it didn't grow. We just don't get enough moisture in the fall for that. So this was a 1 year of data in 2022 from that study. This is root rot incidence. So the higher the bar, the higher the level of root rot, and this is the percent of roots exhibiting any kind of root rot at all. And the rotations there along the bottom, and the year since pea or lentil. So remember, it was pea in 2016 at this field. And so we have 1, 2, 3, and 5 years since it has been planted to a host crop. The letters at the bottom indicate statistical separation. So if it has a similar letter, then those treatments are not considered significantly different. So really the only treatment that was significantly different from the rest was our 5-year rotation here, where we have 5 years since it's been planted to a host crop. Again, it's not no disease, but it is reduced disease. Disease had a strong effect on yield in this trial. It was one of the main drivers of yield, and you can see that reflected here in this 5-year rotation treatment, 5-year break since pulse crops. That's where yield was highest. In our mustard lentil intercrop, obviously lentil yield was much lower. The mustard is going to be competing with the lentil. That's inevitable. But if you have a good market, a good price for mustard, in this case, it was 75 cents a pound in that year. The net revenue per acre was actually higher. Obviously, your expenses might be different, so I don't know if profit would have been higher, but you can look at it from an economic perspective if you're able to make good money on both crops. So this is another crop rotation study. This is field pea. This is conducted by Dr. Michael Bunch at the Carrington Research Extension Center. You might have seen some of these data before, but they kind of confirm what we found in Williston, where if you look here in 2020, that 6-year rotation, in this case, he rotated with wheat, barley, canola, wheat, and corn, all non-Aphanomyces hosts, where there was a significant reduction in root rot severity, whereas you don't see that between a 2 and 3 year rotation. In that case, they're rotating with wheat. If you look at yield, you see a significant yield increase from extending that rotation. And this is in a field with heavy root rot pressure. So achieving 47 bushels per acre under root rot pressure, that is an economic yield in that area. Something like 10 to 20 bushels really is not. So that's comparing a 2 to 4 year rotation, just not as strong as of an effect as a 6-year rotation. So if you have no history of disease, a 4-year rotation might be sufficient. But if you have a lot of root rot pressure in that field, you need to be looking at extending that rotation. So these are data from the Hedinger Research Extension Center. So John Rickerson manages the agronomy program there, and this was done in collaboration with Dr. Michael Bunch. This was a field that had no history of pulses. And on this graph right here, you're looking at root rot severity over time in the 2-year crop rotation with pea and wheat. And as the number of times that the peas have been implanted in that field goes up, root rot goes up. So you can see how disease builds up over time in that field, and actually pretty rapidly if you're considering the lifespan of a farm operation. When you look at root rot severity, again, you see the same trend where you need to be going out to a 6-year rotation to significantly reduce the root rot. In this case, you're not seeing a yield impact yet of root rot, yields are generally a little bit lower in Hettinger. It's drier compared to Carrington, so there might be just less effective root rot on overall yield during that growing season. But with time, that pathogen is going to build up in the soil, and you will start to see the effect on yield. So knowledge is power, and there are a lot of things that can cause lentils and peas to die. And so it's really important that you figure out if you have Phantomyces in a field. So you can now submit soil and plant tissue for testing. This is new in the United States. This is not new in Canada. They've had these testing services for a while, but the National Agricultural Genotyping Center in Fargo developed an assay to test for Phantomyces eutyches and Fusarium avinaceum, Fusarium acuminatum. Those Fusarium species also cause root rot, but the most actionable result is whether or not you have a Phantomyces. And it doesn't necessarily matter how much. If I can detect it in a test, that means you have a significant amount in that field, because it is very hard to detect. We're looking at huge fields here, and when you consider where you're sampling the field, you've got to find the right spot. So that's something we've been evaluating, how you test for soil-borne diseases. So I was sampling 5 locations per field, submitting those separately for analysis, so not submitting a composite sample. And I sampled based on productivity zones. So if you do prescription mapping in a field, your red, orange, and green, red, yellow, and green areas of the field. Generally speaking, where we are, that tends to correspond with topography. So hilltops, side hills, and low spots. Obviously, that's going to be where low spots are going to be where water accumulates, but I haven't necessarily observed a trend on it's always in the red zone, it's always in the green zones, but it can help you identify different zones in your field to sample. If you have yield maps or you have knowledge of the field, you're going to want to go to areas where you've seen issues before. So we tested in the fall because that's when we do all our soil testing. And then I went back in the spring to test roots and see if those test results in the fall correspond to what we saw in the spring. And here in this chart, the red is just areas where we found a Phantomyces, and gray is we didn't. And you can see we had 4 fields in the fall where I identified a Phantomyces from soil samples. That following summer, I detected that in 3 of 4 of those fields in the roots, or detected a Phantomyces in the roots. But there were a lot of fields where I found a Phantomyces in the roots where it wasn't positive in the soil in the fall. So the utility of this test and how we use this testing to predict root rot severity and disease is kind of ongoing. But if you're not sure what's going on in a field, or you want to have a rough idea of which fields to plant into peas or lentils, this is one tool in the toolbox. All right, so let's talk a little bit about variety selection. If you're a new pulse grower, or maybe new to one of these crops, be aware that breeding programs have invested a lot of effort into developing resistant varieties, and that effort is ongoing. So you're not going to want to pick a variety and stick with that for 10 years. It behooves you to stay on top of new releases and to pay attention to their disease resistances. Sometimes that information can be hard to find. You have to do a little digging. But if you're looking at if varieties that are developed in the Canadian breeding programs are adapted to your growing conditions, I really like this Sask Seed Variety Grain Crops Guide. So you can use that QR code. It's a great publication that comes out every year. A lot of information in there, but it also includes all the disease resistance traits of those varieties. For other breeding programs, it might be harder to find that information, but generally speaking, North Dakota State University, Montana State University, USDA-RS, all have breeding programs. Those variety release information that comes out in the publication should describe their resistance to different diseases. That said, scales and level of resistance might differ among breeding programs. Something that might be considered moderately resistant, or MR, in one program. If you have another, maybe a private company releasing a variety, they might call it MR. Those two MRs might not be the same thing. So that's where you might want to go to variety trial data or something like that to compare the level of resistance. Similarly, pathogen populations might differ across different growing regions. So just be aware of that. So in terms of field pea, powdery mildew resistance, we still have people growing powdery mildew susceptible varieties. Sometimes they're in a contract where they have to grow a certain variety. And if this disease occurs, it can be really problematic. It not just causes yield loss, but it also can clog up the combine, and there can be fire hazards from powdery mildew. So if you're able to grow a powdery mildew resistant variety, that's an easy choice. I would always steer people towards that. It's just one less thing to think about. Different varieties might be rated moderately resistant or susceptible for ascochyta blight. There's varieties that have seed-borne mosaic virus resistance. That virus is seed-borne, so that might be something to seriously consider. Fusarium wilt, Fusarium root rot, and I'll talk a little bit about what we've seen for Aphanomyces root rot. So talking about prevention, right? If you don't have any other tools in the toolbox, like a fungicide seed treatment or a foliar fungicide, you're really going to want to pick a variety that has the best resistance package you can find. Chickpea, we're really thinking about ascochyta blight. There's nothing out there that's completely resistant. They're working on it, but this one is where you can't rely completely on resistance, but you should choose the most resistant variety you can find. And then in terms of lentil, some varieties that we grow are very susceptible. We grow richly lentils that doesn't really have any disease resistance, but there are varieties available that have some level of resistance to ascochyta blight, Anthracnose race 1, and Stemphylium. So especially for ascochyta blight, Stemphylium, that might be something to think about. Again, one less thing to manage if you've got some level of resistance. So I'll talk a little bit more about the Aphanomyces tolerance. This is data from Dr. Michael Bunch at the Carrington Research Extension Center. This AAC Julius yellow pea came out a few years ago. I looked at it when I was at NDSU, and then he started looking at it in his trials. And he observed that even under high levels of root rot pressure, he was still getting much higher yields compared to other varieties. So, and then later on, Iron Horse came out, and he observed something similar with this AAC Iron Horse variety. So here under low disease pressure, it's compared to the variety Profit, out yielding that variety even under low disease pressure. But as disease pressure increases, you can see it's maintaining yield a lot better compared to AAC Profit. Obviously, once yield or disease pressure gets up really high, yield is going to collapse because it's not resistant, it's tolerant. And this is just showing yield versus disease pressure here. Again, you see the same thing, that it maintains yield for longer, out yielding Profit, and he's comparing to other varieties as well, but Profit right here is a susceptible comparison. And I just want to emphasize that this is tolerance, this is not resistance. So these roots are still getting infected, and so you're still getting proliferation of the pathogen on that plant. This cannot shorten your rotations by planting a Julius or an Iron Horse. So you see here, this is percent root rot. It's not distinguishable from Profit. And if you submit these roots for testing, which Dr. Bunch did, same levels of pathogen in the roots. So again, it's just able to yield in spite of being infected and having diseased roots. Just this past year, Dr. Bunch conducted a series of on-farm testing, again, including the Julius and the Iron Horse in that testing. And he evaluated another variety, PG Greenback. So this is a green pea, and found it was very, very tolerant. to Aphanomyces. So this is a farm that's had a history of Aphanomyces root rots tested on farm, and he confirmed this at other locations as well. Just speaking to the program that's developed this variety, there can be some potential issues with size for commercial acceptability, but if your contract would allow for this variety and you have a history of root rot, this might be something to consider. All right, so I did a little bit of testing with chickpea, Ascochyta blight at the NDSU Research Extension Center. So if you look over here, this is just Ascochyta blight and yield. These 2 years were very dry, drought conditions essentially, and so overall disease levels were really low. But even in that case, you can see statistical differences between these varieties. So the CDC Frontier, ND Crown, Orion, Leader, these were all the most resistant, not completely resistant, but the most resistant, whereas our Royal, Sierra, those had very high levels of disease, even under drought conditions. And you see that reflected in the yield, much lower yields. These plots were not sprayed with a foliar fungicide compared to the more resistant varieties. So again, for chickpea Ascochyta blight, we want to be planting clean seed and choosing a variety with the best resistance. If you go to that Saskatchewan seed guide I shared earlier, there's some newer varieties that might be slightly better, but overall, right now we're kind of stuck at the same level of resistance. Hopefully in the future, we'll have some improved resistance. All right, finally, I'm going to be talking about planting date. So the concept of using planting date to avoid or prevent disease is based on the disease cycle of our pathogens and what conditions they need to grow and proliferate. So Dr. Porter talked about Pythium in the last seminar, and that pathogen really likes cold and wet conditions. And so when we're planting into cold, wet soils, that's when you have the highest risk of disease. If you have a fungicide seed treatment, that can pretty much take care of it. But if you cannot use a fungicide on the seed, then you really have to look at planting later to avoid that. Well, with peas and lentils, the problem with planting later is that Aphanomyces really likes warm soils. And so if you're planting into a wet, warm soil, you're actually at higher risk of Aphanomyces. So kind of regardless of what soil you're planting into, there's a pathogen that could grow and proliferate under those conditions. But our most problematic diseases, this Pythium and Aphanomyces in our case, right? Aphanomyces on the peas and lentils is what we're really trying to manage, and it likes warm, wet soil. So we want to think about planting early. And so this is data from this chart is prepared by Dr. Michael Bunch, but this data comes from across North Dakota. It's averaged across these locations. We had a few sites in Williams County, and he's had sites in southeast North Dakota as well. So this is averaged across geographic locations. And what you're looking at here is these are soil temperatures at a 2-inch depth, averaged across 7 days after planting. So not just the temperature at planting, but an average across the 7 days post-planting. And as you would expect, given that we know Aphanomyces prefers warm soils, when you're planting earlier under cool soils, you have much less root rot. As you plant into warmer soils, disease severity gets higher. And you can see that reflected in yield, where you're maximizing yield and minimizing root rot by planting when soil temperatures at that 2-inch depth are under 50 degrees. Obviously, you don't want to plant too early. In one trial we had with the field peas, our earliest planted treatment actually got some frost damage, and so it didn't yield as well as our second planting date. So you still have to juggle other things, but 45-degree temperature is still pretty cool, and that's pretty early in the growing season. That's like late April for us. So obviously, if you're in a different climate, these dates and timings might be different, but overall planting into these cool soils minimize losses to Aphanomyces. So this is a combination. This is a very complicated chart, but this is a combination of looking at planting date, variety resistance, and then rotation. So this field 17, this data is from 2024, so it had been 3 years since peas or lentils were planted in that field. In field 18, it had actually been, 9 years since peas and lentils had been planted in that field. And unfortunately, if you look at your yield and disease, it looks like that 9 years actually was very similar to the 3 years. So there could be differences in when peas were planted in that field and how much rainfall it got that season. But just so you know, once root rot becomes a problem in the field, your rotation has to be pretty long. And even a long rotation, if you end up in a wet year, you can still get severe root rot. But here, if you're planting a resistant variety and you're planting early, there's a huge yield benefit. So here you have planting early, late April, and choosing a more resistant variety, you're still able to achieve 66 bushels per acre. Whereas if you planted a little later, maybe even mid-May, you're already losing yield. And in this field, you can see it's even more dramatic actually, where yield really starts to drop off, particularly for the less resistant varieties. So managing root rot is really a combined strategy where we need to be planting early, we need to be choosing less susceptible varieties, and have a long crop rotation. Where you can, use a fungicide seed treatment. All right, so then when we're talking about using planting date for managing foliar diseases, we're mainly trying to avoid raining conditions. So I'll be talking about ascochyta blight. This pathogen is rain splashed from plant to plant. And here in North Dakota, we got, some rain May and June, and then usually the rain shuts off in July. That doesn't always happen, but many years that happens where we don't get a lot of rain in July. So if you're able to delay planting, potentially, you could avoid kind of a rainy weather and maybe reduce or eliminate the need for a fungicide application if you're organic, or reduce the number of fungicide applications needed if you're conventional. And with many other fungal diseases, you have the potential that strategy might work. It would need to be evaluated for your growing region, but it's something to think about because most of these foliar fungal diseases need humidity, need rainfall to complete their life cycle and to spread. So this is some data from a recent publication we put out. This is data averaged across locations in Montana and North Dakota, where we had 3 planting dates, one in early May, one May 20th, which is our crop insurance deadline, and one 5 days after that crop insurance deadline, so May 25th. So the second and third dates are pretty close. The first and the second date are about 2 weeks apart. And you can see that delaying planting did reduce disease. So this hypothesis that we want to avoid the rainy conditions held true. We did get less disease under those treatments. That said, these were not very wet years, and so this would need to be evaluated in a wetter year as well to see if that would work. That said, these seeds were fungicide treated, and so any yield or any losses due to any gains due to planting late would not have to do with avoiding pythium. So when you delayed planting in our case, while you did reduce disease, disease was not severe enough to cause significant yield losses, and so delayed planting reduced yield. Other things that we need to manage here are our soil moisture, right? In a low rainfall year, we really need to plant early to make the most of the moisture in the ground if we don't get a lot of rainfall, or plant early enough that we can take advantage of the early season rainfall when we don't get late season rainfall. And so while we did manage disease or improve disease management, we didn't see that reflected in yield. So if you're an organic producer where you cannot use a seed treatment or a foliar fungicide, maybe that loss in yield compared to early planting would be worth that sacrifice, because the losses due to disease would offset that any prospective yield gain from planting early. So something to think about in terms of managing the diseases I discussed, but also potentially evaluating for managing other diseases, foliar diseases that you might have in your growing region. So just kind of to summarize, managing diseases is holistic. You're not just managing for one thing, and you have to think about what you want to prioritize. Maintaining soil health, managing water will keep the crop stress-free and improve drainage, reducing the amount of root disease in a field. High quality disease-free seed is really important for pulse crops. I would really prioritize that, and keep on top of the varieties. They're coming out with new varieties with different resistance traits. Keep an eye on that seed guide and update what variety you're growing. Long crop rotations are important for pretty much every disease that we're managing. With root rot, it's really long. So try to start with a good rotation where you're introducing peas and lentils to avoid significant issues with Phantomyces, hopefully in the future. And then obviously you need to know what you're battling. So if you're new to pulse crops, there are a lot of resources available out there to diagnose. Our pulse crops working group on our website, so it's just pulsecropsipm.org. If you go to the resources tabs, we have diagnostic guides there. They're available online, but also printed copies are available. If you would like printed copies, please get in touch. I can connect you with somebody who has those. And then check out our podcast, Green Pulse Crops Podcast. Lots of interesting content on there, not just disease, but markets, end use, growers. We have growers on there talking about how they manage their pulse crops. So just getting an idea of how people do things in other growing regions. Finally, I'd just like to acknowledge everybody who contributed their research to this presentation or helped conduct the research that was done. Dr. Michael Wunsch is an amazing resource. If you visit the Carrington Research Extension Center website, all his information and data is there, and he has videos where he explains that. So really great resource, and he stays on top of updating that. Dr. Frankie Crutcher, she's based out of the Eastern Ag Research Center in Montana, part of Montana State University. Her and Dr. Marie Dorval were part of that planting date research. Just acknowledge my former team at the NDSU Research Center, which is now overseen by Dr. Edson Encube, if you're interested in collaborating at the Williston Research Center, and Kyle Dragset, who's the farm manager over there. He drove the big equipment for managing that crop rotation study, and it was really helpful in getting that done. That was kind of beyond my capabilities. With that, I'm happy to take any questions.
That is so wonderful. Thank you so much, Audrey, because South Carolina, we just got approval for 5 fill piece out of my program. So
Nice.
yeah. So the breeder seeds are coming along, and that means like we're gonna call you because the new diseases. With that, I'm gonna ask the first question because we are kind of running out of time. What is the threshold for this new piece coming into the South Carolina? Your recommendation for diseases and the purity of the diseases in the certified seeds. Most of these varieties are resistant to powdery mildew. The list of things that you listed for peas, they quite a bit of resistant fusarium. We got good material, but we need to work on the new emerging diseases. And also our growers are now very big, like North Dakota or Montana. And we have a small growers as well. So I wanted to your thoughts on economic thresholds on the seed selection, and also advices on fungicides like the organic seed treatments. We are working with Dr. Lyndon Porter as well. So, but anything new come up. So I wanted to see what you had to say.
Right. So I think it's important to establish economic thresholds in your growing region. So because economic thresholds might also differ based on resistant varieties. The thresholds I showed you, those haven't been tested in Montana and North Dakota. We're just kind of following them because the Canadian prairies are pretty similar to us. But conditions in South Carolina are going to be extremely different. And so the concept is that you have the pathogen on the seed, it infects the seedling, and then it spreads. So seed to seedling transmission is really important, and that might differ based on variety. So your more resistant varieties might have little to no seed to seedling transmission potentially. And so then your threshold could be higher. And so on. So I think actually we need a lot more research on that area, and we could tighten those thresholds. But generally speaking, it's based on the disease severity and then the risk of seed to seedling transmission. So I think it would be helpful to do that research actually.
Exactly. So hopefully we can collaborate with you. One of my researchers, scientists in my program, she's collaborating with our in-house pathologist to get the thresholds for Pythium. Pythium is another thing for our chickpeas, and Dr. Vandermark is helping on that as well. There's a
So I
lot.
should mention that lab in Fargo can test for Pythium.
Great.
Yes, so something to think about, sending soil samples to understand the quantity of Pythium in the soil for a field.
Yes. We have a problem with sending soils from state to state, need to get those permits and things like that. But we found it's difficult. I need to talk to you in person about that. Let's get these questions that there is a question for those who are working with the homeowners. In South Carolina, these areas, we have a lot of legume growers. They do in the home gardens. So small garden areas where crop rotation might mean like moving plants for just few feet. Does that still help with the soil-borne diseases?
Potentially, if you have soil movement on shovels or equipment, or water moves from area to area, probably not. If it's across the whole yard and they don't connect and you clean tools in between using those different areas, then yeah, that could help. Something to consider though is that you need to destroy any residue on the surface. So make sure you remove anything on the surface because that's where the foliar diseases overwinter.
Right. And also some people grow pulses in the raised beds and then they use the same soil. That kind of no, right?
Well, if you grow peas year after year in a raised bed, you're going to find out.
Exactly. So that's kind of no. Okay.
And in a raised bed, depending on the size, you can get rid of it,
Right.
manageable than a field.
Yeah, this question I get all the time from the smallholder growers, like the small ones. So they grow their whatever they're required for their house, so they grow in the small gardens. So, yeah, that's a good one. Okay, here is the question from one of your students. In the Southeast US, it may be possible to plant a pulse in the fall, but the crop must go through cold, wet winter. To what extent might disease conductive conditions matter during the crop establishment was entire winter period. So basically, he's asking whether he could do something in the fall and keep it for the winter.
Yeah. A big issue of planting in the cold soil is then Pythium. We don't see as much damage on lentils, for example. It's really chickpeas that we see the biggest problems with. So peas and lentils, maybe you can get away with it as long as your crop rotation is good. They do see problems in the Pacific Northwest with like pea leaf weevil, can be really damaging. So you're pretty limited if you can't use a seed treatment. If you can use a seed treatment, I would say make sure you do that if you're fall planting peas. But if not, essentially, you're open to the elements. So really high quality seed that's vigorous, has a high germ, isn't damaged. And if you're working on resistance, I would prioritize that because again, you're in the cool, wet conditions. That's what Pythium likes. Maybe it would give you a leg up on the Phenmyces.
Right, right. But
That work still needs to be done.
exactly. So Tristan Lawrence's master's thesis is all about planting dates. And with my experience breeding pulses for South Carolina, it's very difficult to have establishment in the fall, and then pythium and all those root rot and all those diseases coming in with the fall rain, and they'll never come back. So they'll die and during the winter and they won't come back. The other problem we have is the day length. It's getting shorter and shorter, so you won't get the flowers on time before it gets shorter. So it's a disease, day length, and quite a bit of things for us to establishment. Like you said, we have to do more testing on this. So I got one master's thesis, but more to go. So
well, maybe look at planting in some different soil conditions too, because maybe you can make that work in a sandy soil that drains well, and it's just never going to work in a heavy soil that doesn't.
Absolutely. So Tristan did both soils in Colombia, sandy loam, and here is the clay. So we see what you say exactly. I wish you could visit at some point, maybe next year for our growing season. This year growing season is very short because of the dryness and no much of the rain. So with this, I really wanted to thank you for bringing this idea on the disease control and for the new crop. We are very grateful for your knowledge and helping with the Clemson students. Everything you do is absolutely wonderful, and thank you so much.
Thank you.
With that, our next seminar would be actually researchers from my program. There's two PhD students, Mark Dempsey and Nathan Windsor, will present their research outcome or the research update. Actually, Mark Dempsey is a candidate, he's graduating. And then also Dr. Carolina will present the bioinformatics pipeline. So with that, we'll see you May 6th. Thank you so much. Goodbye.